Alright guys, this is your notes video for your Biological Important Molecules lab. In the first video I'll walk you through what the 4 biologically important molecules are and how they go from being building blocks, the monomers, to polymers, and the parts of the test that we're going to run. And then I will make follow-up videos for each different type of molecule and each different test. So bear with me, highlight along with me, and let's get started. So first, organic molecules are those primarily made up of carbon, hydrogen, and oxygen. The common organic compounds of living organisms are carbohydrates, lipids, proteins, and nucleic acids. Each of these macromolecules or polymers are made up of smaller subunits called monomers. The bonds between those subunits are formed by dehydration synthesis. This process requires energy. A molecule of water is removed, which is called dehydration, and a covalent bond is formed between the subunits. So you're taking water out and you're joining 2 smaller molecules together in order to make a larger polymer. If you want to break that back apart, let's say you eat a burger and you've got those proteins and you need to break them into their building blocks, you break it apart using a process called hydrolysis. And hydrolysis means that you're adding water molecules and therefore releasing energy. So let's look at that in a little greater detail on the left here. So here you've got a part of water, which is a hydrogen, and the other half of water, which is an OH group. And those will come off of it and form water over here on the side. And then the 1 electron left that needs to share with something and a 1 electron over here that needs to share will bunch together and form this bond. Therefore, you're linking a short polymer, poly means many, with an unlinked monomer, mono means 1, and you're making a longer polymer. And it's dehydration. Dehydration is referring to the removal of water. Synthesis means to build something. So when you look down here at hydrolysis, hydro means water, lysis means to break. So we take our water and we add it in right here, and the OH from the water will end up on 1 molecule. The H will end up on the other, and you end up with 2 separate molecules, and that makes your monomer and your polymer together. So we have different methods for identifying our organic compounds. There are different tests available, and it's based on the properties of those organic compounds, how they react to those tests. So forensic scientists will take a sample from a crime scene and they'll look at it and say, I don't know what this is, so let's compare it to other knowns. So we know that different organic compounds will react a certain way every time to different tests. There are more fanciful tests, but for the purposes of our lab, we are just going to look at color change when we add different reagents to a mixture. But one of the important things to remember is you have to have controls to compare them to. So controls are important because they reveal the specificity of a particular test. So it shows what this test is specifically made for. If you use a positive control, you contain the variable for which you are testing. So you know, for example, that iodine makes starch, or iodine plus starch makes blue. Okay, iodine turns blue when it interacts with starch. So starch would have to be present for it to be a positive control. You know, if starch is present and you add iodine, you will get blue, so that would be a positive reaction to your test, and you know that it will demonstrate the test's ability to detect what you expect. A negative control, however, does not contain the variable for which you are searching. So you have iodine and you have a solution, but it's just distilled water. It does not have the starch present, so you know that it should not turn blue. If it turns blue, then something's wrong. So it's often just the solvent, which is just distilled water with no solute and does not react in the test. So it's important when you're testing that you have both things, a known reactant that will give you a positive result and a known reactant or a known control, negative control, that will give you no result. So we will use distilled water a lot in our examples, so you know that it should have no results, so you should be able to compare it to. And we will also use things like, we will do an iodine starch test. You know iodine reacts with starch, but do any of our other carbohydrates react with iodine, and why would that happen? So the first molecule we're going to look at are carbohydrates, and we'll do that in the next video.